Thanks for having me here today. So just quick poll. So I know you guys are mostly master's and doctoral students. So who has an economics background? Who has a neuroscience background? One. Who has a psychology background? Okay. Any other big field? Medicine? Okay, great. Any other fields I missed? Social work? Great. Law, very cool. All right, excellent. So today I thought we'd do a little bit of an introduction on socioeconomic inequality, then a primer on neuroscience methods, which I'm glad I included, and then we'll talk about the literature to date on socioeconomic status and the brain. So focusing on sort of four levels of analysis, looking at behavior, brain structure, brain function, and then finally links to achievement and life outcomes. And I think somewhere between part 2 and part 3 is probably where we'll have our break, but we'll see how it goes. I'm also happy to be interrupted. So if something I say is not clear or it sparks a question in your mind, please do feel free to interrupt me. And I think that'll make for a more pleasant experience if we have some discussion going on. Okay, so the human brain has been termed the most complex 3 pounds in the universe. And it's not hard to see why when we consider the fact that we're born with 100 billion neurons or brain cells, and we generate between 250,000 and 500,000 new neurons every minute in the first few months of life. But it turns out that most early brain growth in the first few years of life is not the development of new neurons or brain cells, but really the connections between brain cells. And these brain connections, or synapses, become increasingly complex in the first several years of life. So this slide shows a picture under the microscope of the connections between our brain cells at birth, at 3 months, and at 2 years of age. And in fact, by age 3, we have 1,000 trillion connections between our brain cells. But what I think is particularly fascinating about this process is that early experience shapes brain development. Now, many of you may have heard of the so-called use it or lose it phenomenon, whereby connections that are used frequently are strengthened, whereas those that are not are dropped or pruned. And it turns out that the brain is most plastic or able to make new connections early in childhood. Now, of course, a child's experience varies tremendously as a function of his or her family's social and economic circumstances. And so we can use socioeconomic factors as a lens through which to understand early brain plasticity. Now, a little bit of background that certainly the economists among us are familiar with. What is poverty? So poverty is a number, an income level, defined by the federal government that varies by family size and composition, meaning the number of adults and children in the home. Interestingly, it doesn't vary geographically. So a family of 4 here in Chicago has the same poverty line set for them as a family in New York City, which has the same poverty line as a family in rural South Dakota, even though clearly the cost of living is quite different in those places. Does anyone want to throw out what the current poverty line is for a family with 2 adults and 2 kids? Anyone know? Very nice. It's easy in a room of economists to get the right answer. So $24,000, just over $24,000. And despite the fact that that's quite low, and if you can imagine trying to raise a family of 4 here in Chicago or another metropolitan area with a high cost of living on that amount, that's quite difficult. And yet nonetheless, poverty is quite prevalent, currently affecting around 1 in 5 children across the US. And of course, we all know that growing up in poverty puts children at risk for a host of negative outcomes in terms of their physical health, mental health, and school achievement. So this is just a slide showing the rates of US poverty over time. You can see that for seniors, shown in orange, that rate has progressively fallen, and we'll talk about reasons why that might be, whereas the rate for children in purple has remained relatively steady at around 1 in 5. Okay. Now, when I talk about socioeconomic status, or SES, I'm talking about more than just poverty. I just told you that poverty is strictly defined based on family income, whereas SES, or as I often call it, socioeconomic circumstance, to avoid that status term, is really about more than just poverty. So yes, income is part of it, but we also consider factors like parents' educational attainment, occupational prestige, and recently people have become very interested in subjective social status, or where one sees oneself on the social hierarchy. And we know that when we conceptualize SES in this way, it tends to be associated with a number of important outcomes in terms of children's cognitive development. Things like achievement test scores, grade retention, literacy, IQ, and school graduation rates. And in fact, this socioeconomic gap in achievement tends to emerge early in childhood and then widen throughout the elementary years. So the data I'm about to show you come from the British cohort study of 1970, which followed tens of thousands of children longitudinally in the UK from age 2 to about age 10. Now, of course, you're not going to give a 2-year-old the same cognitive test that you would give a 10-year-old. So on the x-axis, rather than looking at absolute score, we're looking at percentile, or where children performed relative to their same age peers. So I'm first going to draw your attention to children who at age 2 were performing at the 90th percentile, so outperforming 90% of other 2-year-olds who happened to come from socioeconomically advantaged homes. So those kids from higher SES homes who were high early scorers tended to perform above average throughout the course of childhood. Next, I'll draw your attention to kids who were performing at the 10th percentile at age 2, so only outperforming 10% of other 2-year-olds, who happen to come from socioeconomically disadvantaged homes. So those kids from lower SES backgrounds who were low early scorers tended to perform below average throughout the course of childhood. So far, nothing too surprising in what I've told you. A bit more surprising, though, is what happened to what we might term those crossover groups. So next, let's consider kids who also started out at the 10th percentile at age 2, but who happen to come from higher socioeconomic backgrounds. So those kids from higher SES homes who were low early scorers tended to actually rise in their relative rank over the course of childhood, such that by age 10, they were performing at or even a little bit above average. And finally, and most disconcertingly, what happened to those kids who were outperforming 90% of other 2-year-olds, but who happened to come from socioeconomically disadvantaged homes? So those kids unfortunately fell in their relative rank over the course of childhood, such that by age 10, a child's family socioeconomic background is a much better predictor of cognitive development than is early cognitive skill. And again, just a reminder, we're talking about percentile here. Obviously, no one lost absolute skill between the ages of 2 and 10, but it shows you that relative to your peers, there's quite a bit of mobility there that's predicted by a family socioeconomic circumstance. Okay. So this raises the question as to what factors might be contributing to this socioeconomic gap. So we're a relatively small group, so let's throw out some possibilities. What might be contributing to this gap in achievement?
Access to quality education.
Access to quality education, great. So differences in school quality. What else?
Nutrition.
Sorry?
Nutrition.
Nutrition, absolutely. The first one I list. What else? Sorry? What happens in the home? Yeah, so give me ideas of things that could happen in the home that might lead to these differences. Yeah, absolutely. Differences in parent-child interaction, right? Trauma, 100%, right? So differences in medical aspects of medical care, like prenatal care or perinatal complications, differences in drug exposure prenatally, as well as exposure to environmental toxicants like secondhand smoke or lead postnatally. Differences in the home learning environment or early schooling, differences in family stress, which trauma would be part of that. And so to this laundry list and the many others that you all could likely come up with if I gave you a little more time, I would say yes, really each of those has been shown to contribute in part to the link between socioeconomic circumstance and children's cognitive skill. So how do we make sense of that? Well, one way is to recognize that so-called cognitive skill is really too broad of an outcome, right? So traditional achievement measures like IQ or school graduation rate aren't specific in terms of brain function, right? There's no high school graduation nucleus in the brain. Instead, we can be much more specific about asking which particular cognitive skills and corresponding brain circuitry seem to be most strongly associated with socioeconomic factors. And so that brings us to a neuroscience approach. So neuroscience teaches us that different structures and circuits in the brain support different kinds of cognitive skills. Things like language, memory, executive function, or the ability to plan and pay attention and inhibit your impulses, spatial skills, and many others. And so by taking a neuroscience approach, we can ask which core cognitive systems are most highly associated with childhood family socioeconomic factors. And so that's actually exactly the approach that we've taken over the last 10 years or so. And so we're going to go into some of those findings, but first I wanted to just take a step back and give a bit of a primer on the kinds of methods that we use as neuroscientists. I'm sure you've all seen pretty pictures of the brain with beautiful color splotches in the Science Times. And so we're going to talk a little bit about that and other methodologies and what that really means. So broadly speaking, there are sort of two main categories of methods that neuroscientists use. One set of methods that measures the function of the brain. So that includes things like behavior, right? Simple pen and paper or computer tests measure the function of the brain. EEG and ERP, we'll talk about what those mean. PET scanning, we won't talk about too much. It involves radioactivity, so it's not used very much in kids. FMRI are those pretty brains with the colorful splotches, so we'll talk about how people actually derive those figures. And then the other broad category of neuroscience methodology involves looking at brain structure. So what does it physically actually look like? So the first one is what is it doing? The second is what does it look like physically? And so structural MRI is used for that, as well as DTI, which is basically MRI of the white matter tracts. And so we're going to talk about those different methodologies. So the first methodology is probably the easiest to understand. So neurocognitive behavioral measures, measures that are designed to test behavior, but tap into one or more neuroscience circuits preferentially. So this takes the form of paper and pencil or computer testing. It tends to be relatively inexpensive, certainly in comparison to other types of imaging. And one benefit of neurocognitive testing is that in some cases it can actually be pretty similar to actual classroom activities. So it's perhaps a bit more ecologically valid than showing someone a series of words while they're lying on their back in a scanner. But there's no direct measurement of the brain, of course, so we're making some inferences based on past work about what brain circuits are involved. Does anyone know how we know what brain circuits are involved with different types of cognitive tests? Yeah, so how, sort of historically, how do we know which areas of the brain are involved? If I give you a test of vocabulary, how do I know?
With FMRI?
So that's one set of large body of work, right? So we know that normal, healthy individuals tend to recruit a certain area of their brain when doing that kind of cognitive task. And the flip side comes from studies with patients, right? So if individuals have lesions in their brain and are no longer able to do that kind of task, then we sort of have the necessary and sufficient criteria met, and then we can say with some confidence, okay, I don't need to make a lesion in your brain to know that you wouldn't be able to tell me a list of vocabulary words. If I made that lesion there, I can say with some confidence what area of the brain you're using based on this past work. Okay, so can I have a volunteer? Please. If not, I'll just call on you guys. Dana's like, I'll do it. How about my med student? Yeah? Okay, it's not that bad, I promise. Okay, remind me your name again? Jason. Okay, thank you, Jason. So Jason's going to be our volunteer to do a Stroop task, which is a cognitive test that engages the prefrontal cortex, so this part of your brain right behind your forehead. So if you could just read across the colors of those X's there.
Purple, yellow, red, black, red, green, red, yellow, orange, blue, purple, black, red, green, orange.
Great, good job. Okay. Now, same thing, can you just read those? First we'll start with just read the words, please.
Okay. Purple, yellow, red, black, red, green, red, yellow, orange, blue, purple, black, red, green, orange.
All right, great. So we've confirmed that Jason knows his colors and he knows how to read. Good. Now, the thing that everyone was groaning about, can you please read out loud for us, or say out loud for us, the color of the font of those words?
Sure, here we go. Red, blue, green, yellow, green, black, blue, red, yellow, black, yellow, blue, green, red.
Awesome. Yay. Hand for Jason. Good. Great job. So you didn't make any errors, but you were a little bit slower and it felt a little bit harder, right? So that is executive function, or one aspect of executive function, sort of inhibiting the natural response, which would be just reading those words, for some other response that you're trying to get at. And so your prefrontal cortex is heavily involved in that kind of inhibitory control type task. And we know that, again, because individuals who have lesions in the prefrontal cortex can't do that, and loads of FMRI studies have shown us that it tends to be involved in a Stroop task or other similar task. Okay, so what are examples of studies, of neuroscience studies that have used behavioral tasks? So for example, my colleagues and I first started out investigating using simple behavioral tasks how socioeconomic disparities related to neurocognitive performance. And we'll really talk about the specifics of this a little bit later in the day. But this just gives you a sense when you look at a group of socioeconomically diverse kids. I'll do this sorry, yeah. You tend to see disparities in language performance as well as memory and executive functioning. Okay, so this was purely a behavioral study. We didn't peer into the brain at all. But of course, we're neuroscientists, so we are interested in directly measuring the brain. So how can we go about doing that? So there are a few different ways. The next methodology that we'll talk about is electroencephalogram, or EEG, shown here on an infant. It basically, and it can be done in older kids or adults too, it basically involves wearing a cap of electrodes. And so we can measure the electrical activity of the human brain by placing these electrodes on the scalp and amplifying the signal. And then we plot this change in voltage over a period of time. So we get a signal that looks like this. Most common clinically, people use this to look for seizure activity, but it can actually be used to measure cognitive abilities as well. So then we use computer algorithms to decompose this signal into oscillations that occur in different frequency bands that look kind of like this. So we all have some amount of high frequency brain activity and some amounts of lower brain frequency activity, and we can look at the relative distribution of those frequencies across the scalp in different locations and in different people. So some of the benefits of EEG, it's again relatively inexpensive, and not invasive, not as inexpensive as paper and pencil, but much less expensive than MRI. It has really outstanding temporal resolution, by which I mean we can see differences on the order of milliseconds, so really on the order of the speed of neuronal firing, whereas FMRI, we're talking about differences on the order of seconds, so much slower. But there's relatively poor spatial resolution. So I can talk broadly about differences at the front of the brain or the back or the sides, but I don't have sort of millimeter precision. Now, EEG alone measures the brain at rest, not while doing any particular cognitive activity. But we'll talk about sort of an application of EEG that does get into more specific cognitive activities. And so although resting EEG isn't looking at brain performance during a particular cognitive task, it can be correlated with performance on cognitive tasks or other characteristics. So for example, looking at that distribution of high frequency or low frequency brain electricity, we know that kids who have learning and attention problems commonly have an excess of low frequency oscillations and a relative deficit of high frequency oscillations. Similarly, we can choose a particular frequency band, let's say high frequency power, which is correlated with cognitive ability, and sort of map it across the scalp and then compare differences in frequency bands between groups. So here's a schematic. So if you can imagine someone lying on their back, so this is the back of their head, this is their nose, their ears. This would actually be an average of a bunch of people. And we're looking at gamma power. So that's high frequency EEG power, and where we see it across the head. And what you can see is that in this group and this group, you see a lot at the back of the head, but only in this group do you see that high frequency power at the front of the head. And it turns out that this is kids without a family history of language impairment. And this group represents kids with a family history of language impairment. So just having a family history predicts EEG power in certain regions. Okay. Similarly, a little bit of work has looked at socioeconomic disparities in the mapping of these frequency oscillations across the brain. And work has suggested that individual or kids from higher socioeconomic backgrounds also tend to have more of that high frequency power at the front of the brain relative to kids from more disadvantaged homes. Now I said EEG is measured while these kids or individuals are at rest, but what about measuring the brain when a person is doing an actual cognitive task? So that takes us to our third method, which is ERP, or event-related potentials. Basically what's happening during ERP is an EEG where the subject is looking at a particular kind of stimulus over and over, such as words or pictures. So then when we show that person multiple trials of the same kind of stimulus, we can average those over a number of trials. It reduces noise from unrelated brain activity, and it looks something like this. And we know that different parts of the waveform are characteristic of different things. So for example, when you first perceive a new stimulus, very quickly, within about 100 milliseconds of the time that you're presented that, you see this deviation known as the N1. Later, if you're asked to actually make a decision, is this the same or different from what you've been seeing? It takes closer to a half a second, 400 milliseconds to notice that difference in neural activity. So what's an example where ERP has been used in socioeconomic literature? Again, we'll talk about the specifics of this study a little bit later, but one example is a group of researchers who had kids listen to 2 different stories, one in each ear. And they were told to pay attention to one and ignore the other, which, as you can imagine, is kind of challenging along the same lines as it's challenging to ignore what a word says, but when you're trying to focus on its font and it's actually a color word. And so what these individuals found was that kids of the more highly educated mothers were better able to suppress that distracting stimulus on a neural level. And again, we'll get into sort of the specifics later, but I'm just showing you this now to give you a flavor of how questions about socioeconomic disparities could be addressed using this methodology of ERP. Okay, so I showed you these squiggly lines that are the output of EEG and ERP, but those don't look like brains at all. So what about things that you've actually seen before, taking us to MRI, which gives us these really beautiful pictures of how the brain actually looks. So it involves sitting perfectly still in a dark and noisy tube, which is fine for adults or older children, not so great for the youngest kids among us. So generally, we don't use MRI, either structural or functional, much below 5, really very rarely before 4 or 3. If a child needs an MRI for clinical reasons and they're too young to hold still, they actually would get sedated. So certainly we wouldn't want to sedate kids for research purposes, so we don't do a whole lot of MRI in the toddler years. So how does MRI work? Well, all of our atoms in our body spin in random directions like tops around their individual magnetic fields. But the MRI scanner is a great big magnet, and so when we position our bodies inside the scanner, the atoms in our bodies line up along the magnetic field of the scanner. But there are some unmatched atoms, and so a radio frequency pulse is applied, which is what makes the loud sound, and then the unmatched atoms actually spin the other way. The radio frequency is then turned off, and the extra atoms return to their normal position, which emits energy, and that energy sends a signal to the computer, which uses a mathematical formula to convert that signal to an image. So that's a very quick primer on the physics of MRI. Now, in terms of the characteristics of the methodology, it's a very strong magnet, about 10,000 times the strength of the Earth's magnetic field. It gives us really excellent spatial resolution on the order of millimeters. Again, it's non-invasive in the sense that it doesn't involve any radioactivity. But it is pretty loud. It can be kind of uncomfortable if you're claustrophobic. It's not the most pleasant place to be. Again, using structural MRI, we can we're not looking at what the brain is actually doing, we're just looking at how it looks, but we can take those anatomical measurements and correlate them with either cognitive performance or other characteristics. So to give you an example of how we might use structural MRI in this way, I told you a moment ago that we and others have reported differences, socioeconomic differences in children's memory performance. We've known for 50 or so years that memory is largely dependent on this structure here in green. Anyone know what that is? You guys, someone say it? You guys might remember it from your Psych 101 class if you remember the patient HM who had bilateral lesions in this area, after which point he was no longer able to form new memories. So this is the hippocampus. And we know that the hippocampus is sort of a critical structure for memory formation, for learning and memory. And so we reasoned, and we've also known for quite some time that in many cases a larger hippocampus is associated with better memory. So we reasoned that perhaps, since there are socioeconomic differences in memory, and memory relies on the hippocampus, that perhaps we would see socioeconomic differences in the size of children's hippocampi, and indeed that's exactly what we found. And this study has now, or this finding has now been replicated in 5 independent labs. Okay. The other structural technique we'll talk about is diffusion tensor imaging, which follows the white matter tracts. It's MRI-based and basically measures the location, orientation, and integrity, or so-called fractional anisotropy of the white matter tracts. I think that's probably all we... Okay, and to give you an example of how that's used, in the adversity literature, there are a number of studies that have looked at children who have been previously institutionalized, so reared in orphanages, and kids who have that extreme form of early neglect tend to show differences in the integrity of various white matter tracts compared to kids who have not experienced that. And interestingly, this difference is partially ameliorated following randomization into foster care. Okay. So last technique we'll talk about, what about pretty pictures of the brain that actually tell us what the brain is doing? So again, structural MRI tells us about how it looks, the anatomy. Functional MRI tells us about how the brain is actually working. So how does fMRI work? So essentially, fMRI takes advantage of the fact that blood with oxygen has different magnetic properties than blood without oxygen. So in response to an increase in neuronal activity in a certain part of your brain, your body delivers extra oxygenated blood to that region. And that produces a more intense signal on the MRI. So it's essentially taking advantage of the fact that you have differential blood flow to the different parts of your brain depending on how much you're taxing them with a certain cognitive task. And so we can measure that blood oxygen level dependent or bold signal at thousands of points across the brain. And so what we're really looking for are points in the brain that seem to respond in tandem to the task at hand. So if you can imagine that you have a task where, let's say you want to see which area of the brain is responsible for words. So maybe you would show pictures during the control region and then words and then pictures and then words and then pictures. So you'd be looking for parts of the brain that seem to be differentially active or have more blood flow during the words parts, but not during the pictures parts. Make sense? So we have an experimental condition and a control condition, and we model which parts of the brain seem to vary as a function of which stimulus is being presented. And then we look statistically to see which parts of the brain do that. And so those maps that you see are really statistical maps showing parts of the brain that are statistically more likely than chance to be responding more during the experimental condition relative to the control condition. Okay. So we don't actually have little yellow blobs inside our brain. Okay. All right, so fMRI, like MRI, has excellent spatial resolution on the order of millimeters, moderate temporal resolution on the order of seconds, so much slower than the brain is actually firing because it's following the temporal resolution of blood flow, not neuronal firing. Again, it's non-invasive with no radiation, but it's taking place in the MRI machine, so again, it's loud and uncomfortable. It does directly measure brain function, but the experience of doing a particular task by looking at words and pictures in the scanner might be very different from doing a task in real life. So to take an example from the Stroop task, we could look at which parts of the brain are more active when Jason was reading red, blue, green, as opposed to when he was just reading the colors of the X's. And so that so-called subtraction, or really t-test, gives you a sense of which areas of the brain are more active when you're inhibiting this prepotent response. And it tends to be in the prefrontal cortex. Okay. So how would you use fMRI to investigate questions about socioeconomic disparities or early adversity? Well, one research group has done this by showing that typically developing children tend to show a stronger response in your amygdala, that's this little blob here, when they see their mother's face relative to a stranger's face. That's sort of the typical healthy response. But kids who were reared in an orphanage don't show that difference. So they respond equally to their adoptive mother as compared to a stranger. And that's true even once they're adopted into new homes and clearly do have attachment to their adoptive moms. So suggesting that early adversity can have sort of a lasting imprint on facial recognition. Okay, so that was a sort of quick primer on the different tools in our neuroscience toolbox. Any questions on that?
And sorry if you're going to cover this later. Just let me know. I think I missed a little bit. You said that the hippocampus, was it that if you're at higher income, you form stronger memories?
Yeah. So what I told you was that kids from higher socioeconomic backgrounds tend to have better memory performance, meaning that they're often quicker and more accurate in learning new things that you're then asked to remember. Similarly, they tend to have a larger hippocampus. We will go over that though, and I wanted to give you guys a flavor of the kinds of questions we could ask, but I realized I didn't go deeply into those studies.
Gotcha. And then we will rely that is the case?
Well, we're working on it, yeah, but I'll go over why I think that's the case.
And then secondly, really quick, you talk about kids with severe early neglect, like foster care, they have less white matter, and I might have missed it. So what does that mean?
Yeah, it's a great question too. So they seem to have less white matter integrity. The white matter tracts in your brain sort of serve as connections from one area to another. And so people are working on trying to understand the cognitive associations and what that might be the underpinnings of. We're not going to talk a whole lot today about early institutionalization. I more sort of wanted to give you a flavor of the kinds of questions that could be asked using those neuroscience techniques.
The amygdala and hippocampus combination being the difference between an immigrant student who has a wonderful memory of their past, or a sibling who had like really dysfunctional memory of their past, and just the fact that maybe they had this experience but their memory of it, it's emotional with the amygdala, but memory with the hippocampus, would those be like the two areas that are most connected for that?
So very hard to answer in the case of individuals, right? Almost all of them, we'll go over this point a few times today, but almost all of this data relies on sort of group averages, right? And so I can know something about how, on average, a certain experience puts a child at risk for X or Y brain or behavioral outcome, but I can't say with a whole lot of precision what would happen to an individual child who has that experience, because there's almost always heterogeneity there. Great questions, you guys. Okay, so thought we'd actually launch into some of the substance now. So first of all, what can we say about socioeconomic disparities in children's behavior, particularly brain-based neurocognitive behaviors? So I showed you this slide earlier. This is data from a study that we did looking at 150 New York City first graders who were recruited from socioeconomically diverse backgrounds. We actually did a similar study in kindergartners and on up through adolescents. The idea being that again, we knew at that point that there were dramatic socioeconomic disparities in broad outcomes like IQ or school graduation, but we really didn't have a good sense as of yet as to how those might map onto neurocognitively specific domains, so cognitive domains that map onto particular brain circuitry. And so we simply recruited several different socioeconomically diverse cohorts of families with young kids and administered a bunch of computerized and behavioral tasks that were designed to preferentially tax one brain circuit more than another. Now, with any task, your whole brain is involved, but there are some that are, differentially taxing one system more than another. And what we found in both the data I'm showing you as well as in those other studies was that we tended to see the most dramatic socioeconomic differences in language development, with more modest differences in memory and certain aspects of executive functioning having to do with inhibitory control. And so this pattern seemed to hold from kindergarten on up through adolescence. And then, so all of our work has focused on kids in the US, but another group not so long ago asked a similar question in the country of Madagascar, which is a very poor country. 68% of the country live below the international poverty line. The gross national income per capita is only $340. And they, in Leah Fernald's paper, she recruited a very large sample of over 1,200 kids between the ages of 3 to 6. And it turned out it was actually a nationally representative sample of these kids from Madagascar across different communities. And so she administered the same types of tasks that we had done here in the US, and she found similar socioeconomic gradients. So although all of these kids would meet our definition of poor, there were nonetheless gradients, and so she could look at kids in the lowest quintile on up to the highest quintile of this low-income sample in this developing country. And she found socioeconomic gradients here. I've circled for receptive vocabulary, but it was true also for other domains that we had also previously found gradients, like working memory, which is an aspect of executive function, as well as memory. So it seems that even in a low-income sample, socioeconomic gradients exist in similar domains. Now you might say, okay, well, how early are these effects detectable, right? Oh yeah, go ahead.
Sorry, the previous slide, what is fluid reasoning?
So that is a measure, I believe that they used the Raven's matrices. So it's sort of a measure of cognitive flexibility. Okay, So you might ask, how early are these effects detectable? So our first study started with kindergarten. Clearly, if you're already seeing disparities by the time kids start formal schooling, they must emerge earlier, but when? So in a study that we did a few years ago, we recruited a sample of socioeconomically diverse families with infants between the ages of 9, 15, or 21 months. And we followed them longitudinally. And then administered some commonly used tasks of infant language and memory development to this sample. This pattern you might recognize somewhat from the first slide that I showed you in the British cohort study. So here on the y-axis, instead of percentile, we have z-score, meaning that a score of 0 means that the kids were performing at the mean for that age. And so in blue, we have kids from the most highly educated parents in the sample. In red, we have kids from middle educated parents in the sample. And in green, we have kids from the least educated parents in the sample. And so what you can see is that from 9 to 15 months, we're not really seeing the emergence of socioeconomic disparities in language development just yet. But from 15 to 21 months, what we're seeing is that kids from the more highly educated parents are rising to the top of the distribution, whereas unfortunately, kids from the less educated parents are falling to the bottom of the distribution. And this difference at 21 months of age between the blue line and the green line is not just statistically significant, but also what we would term clinically significant. So it's a difference of about 0.8 standard deviations, which, to put that in context for you, is equivalent in magnitude to 12 IQ points. Now again, this wasn't an IQ test, it was a test of several different language abilities, but it nonetheless gives you a sense of just how dramatic the language disparities already are before kids even turn 2. And we found the same pattern when we looked at infant memory development. So again, by 21 months of age, we found about 0.8 standard deviations of difference between the kids from the most socioeconomically advantaged backgrounds compared to the least advantaged kids in the sample. Work by Anne Fernald's group at Stanford has taken a deep dive into socioeconomic disparities in toddler language development. So she's actually followed a group of kids longitudinally from age 18 to 24 months, which of course gives us sort of a better, we can make better assertions about development if we follow the same kids longitudinally. She's looked at 2 SES groups. Her lower SES kids, I'll point out, are not that low, right? The average maternal education is 13 years, so that's still some college, whereas the higher SES kids in the sample had a little bit more than college. And she administered a series of language measures at 18 and 24 months of age. One was the MacArthur-Bates Communicative Development Inventory, which is simply asking moms you're showing moms a list of vocabulary words and asking if their kids know those words. The other, though, is a much more in-depth paradigm called the licking while listening paradigm, which looks like this. Basically, the kid is sitting on their mom's lap, and they're shown 2 screens, and the experimenter says, "Look at the ball." And then using video recording, they follow where the kid looks. Now, for you or I, for fluent adult English speakers, by the time I say, "Look at the ball," you're already on your way to that ball, right? So you need very little of that stimulus to be able to process and know where I'm going. Kids take a little bit longer, but the speed of that language processing can be very predictive. So what she found was that at 18 months of age, kids from the higher SES group tended to have a higher vocabulary, have more accuracy with where they looked first, and to look faster. And in fact, she found a 6-month age gap by 24 months of age. So here, the word is doggy. You can see at the very beginning, kids are still sort of hearing it, but then sometime around, let's say, a second and a half, you have kids performing it about where they're going to be. So the 2-year-olds, the 24-month-olds from the more advantaged homes, almost all of them, like 85% of them or so, by a second and a half are looking at that doggy. Here, the 18-month-old kids from the lower SES backgrounds, so the younger kids, only about 60% are looking at the doggy by a second and a half. What I think is so interesting, though, is this group here, there's almost complete overlap, right? So those 24-month-olds from the disadvantaged homes are looking just like 18-month-olds from the higher SES homes. So a 6-month gap between 18 months and 24 months. Okay. And it turns out both speed and reaction time are associated with kids' vocabulary levels. Yeah. Can we intervene to change them? Yeah, So people are doing work like that right now. So you'll hear from Dana Susskind in a couple days to talk about the kinds of intervention that might lead to differences in language development. Our best guess is that this is plastic, right? So we haven't gotten all the studies yet to be able to answer that definitively, but we believe this largely has to do with differences in children's experience that could be changed. And we'll talk about what some of those differences might be. So these differences in behavior are dramatic and they appear early. Are people just sort of predestined to their fate based on their family socioeconomic circumstance, or are there differences in experience that could be accounting for these differences? So we think the answer is yes, and there's a few lines of evidence for why that might be. So there are a few studies that have looked at adoptive children. And it turns out when you're looking at broad outcomes like IQ, typically about half of the variation in IQ can be explained by the biological parents and about half by the adoptive parents. So there does seem to be some role of experience at play. Similarly, when we're looking at timing, so if you look at holding constant the amount of time you experience poverty, if you experience poverty early in childhood as compared to later in childhood, typically you fare worse. So given that there are differences in outcome based on timing, that would again suggest that experience plays a role here. And finally, and we'll talk about this, there are a number of examples suggesting responsiveness to intervention, which again sort of presupposes an experience-based resilience. And one interesting or very interesting piece of data comes from the work of Eric Turkheimer, who has looked at the heritability of IQ in twins. So to remind you, monozygotic or identical twins share all of their genes. Dizygotic or fraternal twins share half of their genes, so just the same as any other sibling pair. They're no more genetically alike. And so behavioral geneticists can look at groups of monozygotic and dizygotic twins and look at characteristics that they share or don't share and mathematically compute how much variation must be heritable and must not be. So how does this look? So for example, if you have a group of monozygotic twins and there's 100% correlation with a certain trait in those identical twins, so if one twin has it, the other one always has it. And at the same time, if that trait has a 50% correlation in the group of fraternal or dizygotic twins, so if one twin has it half the time the other twin has it, what does that mean about this trait? It must mean it's 100% genetic, right? So if you share your genes with your twin, you will always be in sync in terms of that trait. And if you don't share your genes, then you won't, right? So it'll be 50% in dizygotic, 100% correlation in the monozygotic. So there are not a whole lot of traits like this, but if you have a trait that's 100% genetically mediated, this would be the pattern that you would see in a twin study. Conversely, if the correlation in a group of identical twins is exactly the same as the correlation in a group of fraternal twins, what must that mean about that trait? I think I heard it. That means that trait must be 100% environmental, right? Because whether you share your genes or not has no bearing on whether you have the same phenotype as your twin. Most traits are somewhere in between. They're a little bit of genetically mediated, a little bit of environmentally mediated, but mathematically we can calculate the relative percentages. Okay, so Eric Turkheimer looked at IQ in twins, specifically looking at the heritability of IQ. And what he found was that among higher SES twins, genetic factors accounted for more variation in IQ, whereas among twins from lower SES backgrounds, environmental factors accounted for more variation in IQ. So it looked like this, with SES on the x-axis in each of these and variation in IQ on the y-axis. So when we're looking at genetic variation, among at higher levels of SES, most of IQ can be explained by genetics, whereas at lower levels of SES, very little of IQ can be explained by genetics. Conversely, when we're looking at, we can just consider these together, shared or unshared environmental variation. At higher levels of SES, there's very little variability in IQ that can be explained by the environment, whereas at lower levels of SES, almost all of IQ is explained by the environment. So how do you make sense of this? Well, one way is to conceptualize it such that in a situation of plentiful access to resources, a child's genetics are largely going to determine his or her IQ. However, in a situation of limited access to resources, those resources might really serve as the rate-limiting step, and the child may or may not be able to live up to his or her genetic potential. Does that make sense to everyone? Okay. All right. So, oh yeah, go ahead.
Oh, sorry. Before you were saying that early childhood poverty is worse than later childhood poverty. So once you have people, like a huge amount of people that actually live through early childhood poverty or extreme poverty in those stages of their lives, what can you actually do to improve their later on?
Yeah. It's a great question. So we're going to talk later in the day about possible interventional approaches. The short answer is our best guess is that interventions are probably more cost-effective earlier, but our brain retains plasticity later in childhood and adolescence and adulthood too. So even if interventions are potentially less cost-effective, it doesn't mean that they shouldn't be done. Sure. Okay, so if we can all agree that experience is probably accounting for at least some of these disparities in outcomes, again, which experiences might be explaining these differences. So I showed you this slide before. In my lab, we're particularly interested in two of these, namely the home language environment and family stress. And so I'm going to show you a theoretical model of what I think is going on, and then show you some data throughout the rest of the day to support it or show you how we're testing the model. So the theoretical model goes something like this. We know that there are socioeconomic disparities in the home language environment, meaning the quality and quantity of words that kids hear in the home. In turn, we believe that differences in the home language environment likely have cascading effects on the development of parts of the brain in the left hemisphere that support the development of language. Simultaneously and relatively independently in the literature, we know that there are socioeconomic disparities in exposure to family stress. Family stress, in turn, has cascading effects on several brain regions, notably the hippocampus, which supports memory development, as well as the prefrontal cortex and limbic circuitry, which are important for cognitive and emotional regulation. So I told you that these are the sort of cognitive domains, I think my pointer is dying, where we see the largest socioeconomic disparities. Now we're going to take a step back to talk about what are the brain regions that are likely underlying those disparities, and then what are the experiences that likely are affecting these brain regions. Okay, so that brings us to the second part of the talk, looking at socioeconomic disparities in brain structure. So a few years ago, I had the good fortune of collaborating with the Pediatric Imaging Neurocognition and Genetics investigators, or PING investigators, who had conducted a really unusual and state-of-the-art study at the time, recruiting over 1,000 children and adolescents to do a brain imaging study. So this is a very large sample for brain imaging work, because it's expensive to do. And they recruited kids from a broad age range between the ages of 3 and 21. This is a cross-sectional sample. And fortuitously for our line of questioning, it happened that it was quite a socioeconomically diverse sample recruited from 10 sites across the US. So the PING study wasn't designed to ask questions about SES. It was really designed to give us sort of state-of-the-art information about structural brain development cross-sectionally across ages. But because it was such a diverse sample, we were able to ask questions about socioeconomic disparities in brain structure. So the socioeconomic background of these families was quite broad. On average, parents had some college, but it ranged from parents who had less than a 7th grade education to parents who had an advanced degree. On average, the average household income was certainly above the national average, hovering at around 100,000, but again, quite a broad range from families who were living in poverty on up to many times above the poverty line. And it was sort of a novel study in that it examined various aspects of brain structure. I'm not going to get into this too deeply here, but most commonly, brain structural studies prior to this point had looked at brain volume. If you imagine your brain being the shape of a soup can, if you remember to your high school geometry, volume would be the amount of soup that can would hold, but you would calculate that by multiplying the height of the soup can by the area of the circle on top. And it turns out that the analogous measures in the brain for height of the soup can would be the cortical thickness, and the area of the circle on top would be the surface area. And the problem with measuring volume is that those two measures, cortical thickness and surface area, go in opposite directions over the course of childhood. So our cortices become progressively thinner, so going from like a soup can to a tuna fish can, and the surface area becomes progressively larger, so going from like a tomato paste can to a full-fledged can of soup, right? So if you're just measuring volume, you're kind of missing some of that nuance. So we were able to separately look at surface area and cortical thickness in this sample. The other big innovation in the Ping study was that they measured genetic ancestry, which we can sort of, for this purpose, define as genetically defined race. It's your proportion of ancestral descent from one of six, or from 6 different racial groups. Okay, so what did we find? So on average, in this group of over 1,000 children and adolescents, we found that higher family income was associated with larger cortical surface area. So this is using MRI, so they look more like brains. So to orient you guys, this is the front of the brain, this is the back, this would be a view of the side, the lateral surface. Here again, this is the front of the brain, this is the back, this would be a view straight down the middle, the medial surface. And so every point that you see there in color is a point where, at that point, higher family income was associated with larger cortical surface area across the sample when controlling for things like age and sex and genetically defined race. So what you can see is that this held true across most of the brain, right? You see these income disparities across most of the brain, and yet there are certain areas where you see that this is, that you see a particularly strong relationship, right? So the hotter colors in yellow are the ones where this relationship was particularly strong. And that's interesting because those hotter colors are the very areas that support language and executive functioning. So the brain data recapitulated the behavioral data where we saw the most dramatic socioeconomic disparities in the regions that support the very skills where we had previously found disparities. Now, a couple important points to note here. So this relationship between family income and children's brain structure wasn't linear. Instead, we saw this relationship most steeply among the most disadvantaged children. So that suggests that dollar for dollar, relatively small differences in family income were associated with proportionately greater brain differences among the most disadvantaged families. And that kind of makes sense when you think about it, right? So an extra $10,000 for a family making $200,000 would be nice, but probably wouldn't make a dramatic difference in family life. Whereas an extra $10,000 for a family making $20,000 is a pretty substantial difference. And the second point, this is perhaps the most important thing I'll say today, there was tremendous variation from person to person. So yes, on average, in this sample of nearly 1,100 children and adolescents, we found that higher family income was associated with larger cortical surface area. But there were plenty of kids from more advantaged homes who had relatively lower cortical surface area, higher cortical surface area, plenty of kids from more advantaged homes who had lower surface area, plenty of kids from more disadvantaged homes who had higher surface area. So in no way could I know an individual child's family income and with any accuracy predict for that child what their brain would look like. Does that make sense? Okay. Subsequent work looked at that other measure I mentioned, cortical thickness. So as I said, our brains, our cortices, the thin layer of cells on the outer surface of our brain that does most of the cognitive heavy lifting, tend to get thinner with age, and that by far is the biggest effect that you see for cortical thickness, right? If you squint, all you really see is that it's decreasing with age. But we do see a little bit of moderation of this finding with SES. So families from lower income backgrounds shown in blue show a slightly steeper age-related decline and then a leveling off, whereas children from the higher family incomes show a more gradual age-related decline in cortical thickness. There is some evidence both from the animal literature and from other aspects of early adversity literature in humans that suggests that early adversity actually speeds development. So there may be a hint of that going on here with kids from the lower socioeconomic backgrounds showing a more rapid cortical thinning. But you'd really want to look at that longitudinally to be able to say something for sure. Okay, now you might say, does it really matter, right? Do these differences in structural brain development mean anything, right? I mean, if you're finding these differences under the hood, but they don't translate into differences in cognition, who cares? So preliminarily, we think they do matter. So in this work, links between family income and children's executive functioning could be partially explained by these differences in brain structure. And work by other labs at MIT and at the University of Wisconsin-Madison suggests that differences in brain structure may account for anywhere between 15 and 44% of the income achievement gap. Okay, so what experiences might be accounting for these links? So I showed you this slide before. For the next part of the talk, we're going to focus on that top arm. What can we say about differences in the home language environment and how they might be accounting for these brain developmental differences? So this is work by Hart and Risley, who you'll hear more about in a couple of days. But basically, Hart and Risley did a very interesting experiment in the mid-1990s where they recruited socioeconomically diverse families and followed them longitudinally for the first several years of their children's lives. And every month, they went into the homes of these families and for about an hour or so tape recorded everything that the children heard and everything that they said. They then brought their tape recorders back to the lab where some poor research assistant had to transcribe everything. And what they found was that at every age, the kids from the more socioeconomically advantaged homes tended to hear more words than the kids from the more disadvantaged homes. And so extrapolating or calculating out what that would look like over every waking hour of the first several years of these children's lives, the authors calculated that this amounted to a 30 million word gap in terms of the number of words kids would have heard in their short lives from more advantaged backgrounds relative to less advantaged backgrounds. Now, subsequent work showed it wasn't just the number of words that varied as a function of socioeconomic background, but also the complexity and responsiveness or back and forth conversational nature of the interactions that varied as a function of socioeconomic background. And subsequent work to that showed that the number of words that the kids heard was directly related to their vocabulary size, perhaps not so surprising given that huge disparity in experience. Now, I just showed you that one of the regions in which we see the most dramatic socioeconomic disparities in brain structure are the very regions that support language development. And so we're asking right now in my lab whether differences in the home language environment might be explaining socioeconomic differences in brain structure. We have it a little bit easier than Hart and Risley and their poor research assistants. We're able to use this little device known as the LENA, or Language Environmental Analysis tool. It's basically a small digital recorder that fits into specialized clothing that will automatically record for up to 16 hours. Then, assuming that the family gives it back to us, we can plug it into our computer and the software automatically analyzes things like the number of adult words the child hears, the number of vocalizations the child makes, as well as the conversational turns or this back and forth nature of conversational interactions. And what we're finding is that more conversational turns are associated with greater surface area in left hemisphere language cortex. So that's showing you that here. So more conversational turns, kids have more surface area in this region known to be responsible for language and that we've also previously found socioeconomic disparities in. And in fact, this difference in surface area in this sample partially mediates socioeconomic disparities in reading skills. I should say this is a group of 5 to 9-year-old kids. What's really interesting, at least to me, is that while we see this link between brain structure and conversational turns, we see no link between brain structure and either the total number of adult words or the total number of child vocalizations. So it's not just like lots of talk going around the kid that matters. It really does seem to be something about the back and forth responsivity of the conversational turn. Similarly, it's not the case that kids who are talking more and yammering more are either having differently shaped brains or even seem to be engendering more conversation. It really does seem to be something intrinsic to that responsivity. Okay. So what can I tell you about the bottom arm of the model? What do we know about socioeconomic disparities in family stress? So first, I thought we would talk a little bit about what is stress, particularly from the neuroscience perspective. So typically, we define stress as a novel or threatening situation that increases heart rate, blood pressure, and stress hormones like cortisol. It turns out, though, that some stress is actually a good thing, right? So you wouldn't want your entire life to be stress-free. You probably would not develop appropriately if that were the case, even though it might sound good right now. But really, there are certain forms of positive stress. So for example, these are stressors that are mild or moderate, tend to be relatively brief, over which the individual feels that he or she has some control. And importantly, these are stressful experiences that tend to be buffered by healthy relationships. And clearly, learning to adjust to this kind of positive stress is really important for healthy development for children. So some examples might be dealing with frustration, meeting new people, getting a shot, brief separations from parents, right? All of these challenges are things that kids need to learn to navigate in the world. So moving on from those kinds of positive stress that everyone agrees are, generally an important part of life, we can think about tolerable stress. So these are stressors that are more severe, that do have the potential to negatively impact the developing brain, but if they're generally for limited time periods or occur in the context of buffering relationships, the effects on the developing brain can be reversible. So for example, these would include death or illness of a loved one, a frightening accident, divorce, or any other negative event that might be relatively traumatic, but that occurs in the context of ongoing supportive relationships. Kids are very resilient to handling this kind of tolerable stress. And finally, we can consider what has been termed harmful or toxic stress. So this would be stress that's extreme, long-lasting, frequent, and importantly, for which buffering relationships are not available. And we know that this kind of toxic stress can lead to damage of both the body and the brain. It can result in poorly controlled stress response systems, where either the stress response system sort of shoots up at the tiniest little stressor, or in which the stress response system really seems to burn out and doesn't really seem to respond appropriately to events that should be stressful. So it can, again, be either overly reactive or slow to shut down. Children in these instances may experience clinically relevant anxiety or feel threatened even when there's no real threat present. And this can ultimately lead to mental health problems like depression or anxiety or substance abuse, as well as physical health problems like heart disease, diabetes, and stroke. So I think I'm not going to play this video in the interest of time, but I encourage you to check out the Harvard Center for the Developing Child. They really have a number of excellent videos looking at the effects of toxic stress on the developing brain and body. If we have time later, I'll play one of them, but I want to make sure I get through everything and also give you guys a chance to ask questions. Okay, so we know that one sort of key target of toxic stress is the hypothalamic pituitary adrenal, or HPA, axis. So this is sort of the backbone of our natural stress response. Our hypothalamus in our brain secretes a hormone called CRF, which acts on our pituitary in our brain to secrete a different hormone called ACTH. That's then released into the bloodstream and circulates down to our adrenals, which are located in our backs right above our kidneys, and our adrenals secrete cortisol. So that cortisol helps us do lots of things in reaction to stress, and then that cortisol that's being secreted in the blood then serves as a negative feedback and turns off the hypothalamus. That's how it's supposed to work. And so when it's released suddenly and turned off quickly, it causes us to mobilize our energy, it enhances our memory, it activates our immune response. We're sort of ready to do that fight or flight mechanism. But if stress is chronic and cortisol release is dysregulated, our immune function can end up being suppressed, so you can get sick more. Our memory can be suppressed, so we have trouble learning. And it can lead to a number of physical problems, like metabolic syndrome, which is sort of a precursor to diabetes, bone loss, and muscle atrophy. So stress is bad for you, toxic stress anyway. And it turns out that there are 3 brain regions that really tend to be dramatically affected by stress. And we know this from both the animal literature as well as some aspects of the human literature. And these are the hippocampus, the amygdala, and the prefrontal cortex. So I think we'll focus on the effects of stress on the hippocampus for now. If we have plenty of time left over at the end, I can go back and, if people are interested, talk about the effects of stress on the amygdala and prefrontal cortex. Okay, so again, what is the hippocampus? We've talked about it a little bit already. It's essential for memory. If you have a lesion in your bilateral hippocampus, you really can't learn and remember anything new. It's also essential for navigating spatially around the world. And critically, for our purposes today, it has very high levels of what is known as glucocorticoid receptors. So these are receptors that are responsive to circulating cortisol. Okay, so how does stress influence the development of the hippocampus? So this has been heavily studied in the animal literature, where researchers can actually either provide stresses to animals or provide stress hormone to animals and then dissect out their brains and take a look at the hippocampus. So it clearly involves experimental paradigms that would not be possible in humans. And so we know when researchers apply stress or stress hormone to rodents, it suppresses neurogenesis in the hippocampus, so the hippocampus doesn't develop new neurons at the same rate. It alters the activity of neurotransmitters, so it alters the cellular function in the hippocampus. It changes the cellular structure in the hippocampus. It leads to overall smaller hippocampal volumes, and it impairs learning tasks that are dependent on the hippocampus. So the hippocampus is sort of a prime system that really seems to be responsive to high doses of stress in animals. One rodent model of stress and maternal care is licking and grooming. So believe it or not, mouse mothers rodent mothers lick and groom their pups. And they vary in the amount that they do this. Some mothers are naturally high lickers and groomers. They spend a lot of time engaging in these behaviors with their pups. And others do it much less frequently. And so it turns out that the offspring of the mothers who don't lick and groom as much show increased responses to stress, as well as enhanced emotionality. So they sort of respond more vigorously to emotional stressors or anxiety-producing or painful stimuli. And they're less good on spatial learning tasks and object recognition tasks that rely on the hippocampus. So it seems like the offspring of these mothers who aren't engaging in these warm behaviors are doing less well in response to stressful situations. Now you might say, well, maybe that's just, genetically these moms are less likely to engage in these behaviors and their kids are less likely to show these outcomes, and maybe it has nothing to do with the experience. But again, in a way that wouldn't be feasible in human research, researchers can actually cross-foster rodents, right? So they can take the offspring of a low licking and grooming mom and put them over in the cage with a high licking and grooming mom. And it turns out that in that case, if that offspring, that pup, gets the high licking and grooming behavior, the so-called warm and nurturing phenotype, they look like the biological offspring of that high licking and grooming mom. Right, so these effects are recovered. So when we actually look at the hippocampus under the microscope, or other people look at the hippocampus under the microscope, maternal care affects the morphology or structure of the hippocampus. It affects long-term potentiation, which is like the cellular function of the hippocampus, and it affects behavioral performance that relies on the hippocampus. Now, what about in humans? Clearly, we can't do these kinds of experiments where we induce stressful situations and then, move people to different families. But we can ask on a correlational level, how is stress associated with hippocampal structure in humans? Again, in people, you're not going to be able to measure things at the level of cells, but we can measure the hippocampus by using MRI, right? So we can look at the size of it. It's much more coarse than looking under the microscope at the individual cells, but it's what we have at our disposal in human neuroscience work. And it turns out that higher perceived stress is associated with smaller hippocampal volumes. So this was a study that followed a number of individuals longitudinally, I think for 20 years, and they looked at their average perceived stress. They just had them rate on a scale, I think of like 10 or so questions, different aspects of their daily stressful life. And the individuals who rate their stress more highly had a smaller hippocampus. Now again, it's not the sort of quality of research that you could do in the animal world, right? Because you're not randomizing people to anything. You can't randomize people to different levels of stress. But it's at least correlational work that's consistent with what the animal literature would have predicted. What you can do in human work is adjust for a number of potential confounds, and even when they adjusted for everything that they could think of, this effect still held. Other human studies have been consistent with this, suggesting that acute and chronic stress can impair the function of the hippocampus. There's some evidence that stress may reduce hippocampal volume. That's been shown in studies of individuals with post-traumatic stress disorder or PTSD, as well as healthy middle-aged and older adults, and even some evidence in children. But, and here's a really good example of where correlation is not causation, perhaps it's not the case that stress is leading to a smaller hippocampal volume. What if it's the case that a smaller hippocampal volume causes you to perceive stress differently? So this interpretation came from a study a few years ago that looked at twin adult males, one member of whom was sent off to combat, I think it was the first Persian Gulf War, and the second member of whom stayed home. So these were identical twins, but again, one was exposed to the atrocities of war and the other was not. It turned out that the likelihood for the twin who went into combat to develop PTSD was predicted by the size of the hippocampus of the twin who stayed home. So maybe it's not that, going to war or having some other trauma shrinks your hippocampus. Maybe it's that a smaller hippocampus predisposes you to experience stress differently. So the jury's still out. I think the preponderance of evidence probably does suggest that stress leads to a smaller hippocampus, but I think we don't have it all clearly worked out yet. Okay. And again, if people are interested, I'm happy to sort of talk about the effects of stress on prefrontal cortex and the amygdala a little bit later on. All right, so that was sort of a long period of time talking about this link here, right, between family stress and the hippocampus. But here we're fundamentally interested in socioeconomic disparities. So what can we say about socioeconomic disparities, stress, and the hippocampus? So socioeconomic disadvantage is associated with higher family stress, as you might imagine. So caused by things like limitation in material resources, chaos in the home, sometimes exposure to neighborhood violence. And we know that when parents are distracted or depressed, their interactions with their children are more likely to be characterized by conflict or withdrawal, rather than the kind of warm and nurturing parenting that we know is really important for children's development. Work by our group and others have suggested that socioeconomically disadvantaged children may have altered levels of stress hormones like cortisol. And I just showed you that cortisol can have cascading impacts on the hippocampus. I didn't show you, but I can show you later if you're interested, that it also has cascading impacts on the development of the prefrontal cortex. And so, as I mentioned before, we've previously found socioeconomic differences in children's memory development. We reasoned that would be, therefore we would find socioeconomic disparities in the size of the hippocampus, which is what we found. So given that there are socioeconomic differences in the size of the hippocampus, and given that stress has cascading effects on the hippocampus, would chronic stress explain these socioeconomic links with the brain? So either the hippocampus or with the prefrontal cortex. So that's work that we're doing right now. In our lab, we're measuring cortisol using hair samples. If any of you are familiar with the literature, you know that more commonly it's measured using saliva. But it turns out, so salivary or blood levels of cortisol start out high at the beginning of the day and then decrease over the course of the day. So that to really measure it correctly, you need to take several different samples over the course of the day. It's also very sensitive to different things that are going on in the individual's life. So if the person's in a bad mood, or the kid just threw a temper tantrum, or even is on certain medication, or ate something different, cortisol levels in saliva can be affected. And so typically, you would measure it several times over the course of a day, and maybe even over several days. So I don't know about you guys, but if someone handed me 9 swabs and said to measure my kids 9 different times over the next few days and to be very careful about when it happened, I would probably mess up. Whereas cortisol is chronically secreted into hair. So by taking a small measurement of it, we can get an average level of cortisol over the last several months. So we do it sort of one time when they come to our lab. And so what have we found? So we are seeing socioeconomic differences in hair cortisol with kids whose parents have more education. This is parent hair cortisol. So parents with more education tend to have lower hair cortisol. And we found the same thing for their kids. And importantly, kids whose parents have more education have lower hair cortisol, even when controlling for their parents' hair cortisol level. When we looked at the link with income, here we found a nonlinear relationship. So suggesting that at the low income level is where we see the biggest difference, similar to the brain data I showed you, right? So for parents, small differences in income are associated with proportionately greater drops in hair cortisol at the low end of the income spectrum. And interestingly, we have not found links between family income and children's hair cortisol level, so suggesting that it may not be material resources so much that's leading to differences in kids' stress. Now what about the effects on the brain? This is some pretty recent work that we just wrote up, but again, finding that higher levels of hair cortisol in the kids is associated with a smaller hippocampus. So this sort of supports our overall theory that higher stress is having a cascading effect on the hippocampus in the direction that we would expect, leading to a smaller hippocampus. Okay. And if anyone's interested, it's specifically in the CA3 and dentate gyri, which are the regions that have been found to be most sensitive to stress in the animal literature. Yeah.
Do we have any pointers as to what aspect of socioeconomic status affects cortisol? Is it like the time spent?
It's a great question. So no, that would probably be the next level of inquiry. What is the aspect of socioeconomic status that's leading to differences in cortisol? I mean, we ask a lot of questions about perceptions of stress. Well, we ask the parents a lot about perceptions of stress and life events they've experienced in their own mental health. We have a little bit on children's mental health, but it's harder, as you might imagine, to get good responses on stress from young kids.
To kind of follow on that, I just want to make sure. So you were finding that income doesn't really matter as much. It's really just about the level of education, and you're defining education as like high school, college, master's.
So we define education continuously. And in that study, it was true that family income was not associated with kids' cortisol levels, but it was associated with parents' cortisol levels. And I would caution you to interpret too much from any one study. Like, what we really need is convergence in the literature, and there haven't been enough studies yet to really be able to say. Okay. All right. And so then the final step in asking this question is not just whether socioeconomic background is associated with the hippocampus, not just whether it's associated with stress physiology, and not even just whether the cortisol itself is associated with the hippocampus, but whether there's a statistical mediation there, and it turns out there is. So when you control for child hair cortisol, the link between parents' education and children's hippocampal volume is reduced. Now again, this is really using statistics to try to get very preliminarily at some causal inference, but how would you really want to test this experimentally if you could? So the answer is you'd want to change parent education somehow, right? So what you'd really want to do is see what happens if you could somehow make that happen and randomize people to get different amounts of education. Would that then lead to a cascading effect on children's stress hormone and brains? Okay. All right.
Well, presumably you would want to separate, because presumably education and income are correlated. So you'd also be interested to know, like, if you just gave parents more money, does that also change it? If you give them more education, if you give them more money, like, does it matter one or the other? Or does both actually make a difference?
And we'll talk about that in the second half today. I think one of the papers I had you guys take a look at ahead of time was this paper by Joan Ludby's group, which found something very similar. So this was a pretty large sample of a couple hundred kids, where she found this link between parent income and children's hippocampal volume, and found that it was mediated by parenting and stress. And so again, it's not clear whether it's income, education, or both, but sort of socioeconomic circumstance broadly defined is associated with these differences in the brain, and in the case of the hippocampus, we think it's likely explained by family stress. Okay. Now you might ask whether it's childhood SES or adult SES that seems to be driving brain development. So in one interesting study by Roger Staff and his colleagues a few years ago, they looked at a couple hundred senior citizens, so 64 to 65-year-olds who were born in 1936. And he and his team asked them about what their childhood SES was like so asking them concurrently at 64 what their childhood SES was like. And so, of course, you're not really going to know what your family income was, maybe ever, but probably not, with that long of a delay. So instead of asking things like that, he asked how many public rooms did you have in your childhood home, how many people had to share a bathroom, what was your father's education when you were 11, and then he also measured their adult SES in traditional ways. So the number of years of education that the individual had obtained him or herself, what their occupation was, and a measure of neighborhood SES looking at local area deprivation based on their address. And so what he found was that childhood SES, even measured in this relatively coarse way, was associated with the adult's hippocampal volume, whereas their own socioeconomic attainment was not. So, of course, we can talk about potential for recall bias and whatnot, but even given that, the childhood factors were a better predictor than was their own educational or income attainment. And the last thing I wanted to mention today before we take a break, so we've talked a lot about socioeconomic disparities in stress and adversity and how that might have cascading effects on the brain. Certainly, we really do believe that poverty is associated with early exposure to chronic stress in ways that we believe may be harmful to the developing brain. But the effect that we're talking about predominantly here today is much less severe than the profound harm that would be caused by depriving children of their families, right? So the science is very, very clear. There's been quite a bit of research looking at kids who are previously institutionalized, and that isolating children from families leads to profoundly abnormal health and development. And so it's very clear that nurturing relationships are really absolutely fundamental to normal human development and resilience, and that policy that doesn't support that is very likely to have long-term detrimental effects on kids. So when we're talking about these effects of poverty, they're real effects, but we're still talking about kids who are being reared in sort of species-typical environments and not separated from their parents. So for the first half, we heard sort of broadly about socioeconomic disparities in cognition, did a little introduction about neuroscience methods, then we talked about the research linking socioeconomic disparities to children's behavior, and then brain structure, with a little bit interwoven on the kinds of experience we think are likely mediating those behavioral and brain structural differences. So now for the third portion, we'll talk about socioeconomic disparities in the function of the brain, and then finally we'll link it back to how we might apply this research to interventions. Okay, so in terms of brain function, right, it's not all about how the brain looks, but actually about what the brain is doing. I showed you this question before of just how early are socioeconomic disparities detectable. So spoiler alert, in terms of behavior, we seem to see these differences in the second year of life. So that was the data I showed you in our work, looking at differences between 15 and 21 months of age. In Anne Fernald's work, looking at that 6-month gap in language between 18 and 24 months of age. Interestingly, though, in terms of the brain work, it seems that we might be able to detect differences sooner. And that sort of makes sense if you think about the fact that, behaviorally, it's hard to probe an infant, right? It's hard to measure language development in a child who's not really talking yet. So there are ways that we can try to get at what kids are understanding when they're very young, but it's a bit more direct to measure the brain itself. And it does indeed seem that we see disparities along socioeconomic lines in brain function before we can measure them behaviorally. So again, this was the slide that I showed you earlier, suggesting that the emergence of socioeconomic disparities in language development seem to emerge sometime between 15 and 21 months of age, so in the second year of life. Same thing for memory. And I showed you earlier that EEG measures brain function by measuring the electrical activity at the scalp, reflecting the underlying electrical activity in the brain. And we talked about how EEG measures high frequency and low frequency electrical signals, and that kids who struggle with learning and attention tend to have too much low frequency signal and not enough high frequency signal. And so Dr. Natalie Brito, who was formerly my postdoc, she's now an assistant professor at NYU, was interested in tying these two questions together by asking whether we see socioeconomic disparities in infant brain signal very early in the first few days of life. So she looked at a cohort of very young infants, really just a few days old, measured EEG, and asked whether we would see socioeconomic disparities in brain function, looking at these relative ratios of high frequency or low frequency electrical activity. And the answer was a resounding no. So there was absolutely no evidence for socioeconomic disparities at birth anywhere across the scalp in any frequency band. Now, it wasn't the case that there was no variability in infant EEG power, right? Maybe it would be that they were all looking the same, but that wasn't the case. You can see there is quite a bit of variability there. It's just not explained by family income or parents' education. Now you might also say, well, okay, maybe there's a lot of variability, but maybe it's all just noise, right? Like who knows what a 4-day-old is thinking or what their brain processing looks like, right? So maybe there's variability, but it's meaningless, it's just static. But that doesn't seem to be the case either, because in the same sample, their newborn brain function variability predicted 15-month cognitive development. So it didn't seem to be the case that these differences, these individual differences in brain function at birth were meaningless. They just weren't accounted for by socioeconomic factors, okay? However, in a different sample that we are collecting right now in my lab, looking at infants at 6, 9, and 12 months of age, we are seeing the emergence of socioeconomic disparities in brain function. So this is work that is ongoing, but somewhere in the second half of the first year of life, we are able to detect socioeconomic differences in resting brain function along the pattern, the way in terms of the patterns that we would have predicted. So higher family income is associated with more of that high frequency power in the front of the brain. Did you have a question?
Yeah, what did the frontal gamma EEG color mean? Is it like an averages measurements across time, or is it what EEG was?
So it's looking at the average power of high frequency EEG signal from the front of the brain electrodes. No, this is resting. But that is itself predictive of subsequent cognitive abilities, not just in our work, but in others as well. Okay. Now, we also talked earlier about how ERP is another way of measuring brain function using the same EEG machine, but as opposed to just looking at resting brain function, ERP takes advantage of stimuli by showing the same kinds of stimuli over and over and then averaging across the signal across those stimuli. And so in the example I showed you earlier, we're going to dive a little bit more deeply into it. The researchers, which the lead author was Courtney Stevens, this took place in Helen Neville's lab at the University of Oregon. They asked whether family socioeconomic backgrounds related to a child's ability to pay attention to relevant information and ignore distracting information. So again, in this study, the researchers had the children who were between the ages of 3 and 8 wear headphones where in one ear they heard one story and in another ear they heard the other. Of course, which ear was listening to what was counterbalanced across the children. And they were told, let's say, for example, to pay attention to Harry the dirty dog, but ignore Where Are You, Blue Kangaroo, or vice versa, depending on the child. And so subsequently, the researchers, and these were kids from socioeconomically diverse homes, subsequently they asked the kids questions about the story of interest, whichever one they were supposed to be paying attention to. And interestingly, the kids across these different socioeconomic groups answered equivalently accurately. So it did seem like everyone was getting the information that they needed from the channel they were supposed to be listening to. But there's a bit of a but when we look at the neural processing. So who were these kids? It's a relatively small sample. They all had normal language, and they used maternal education as their SES measure. So in this case, higher maternal education meant at least 1 year of college. Lower maternal education meant no more than high school. You'll see that these definitions vary tremendously from one study to the next. In our work, we tend to analyze them continuously so as to avoid arbitrary distinctions. And again, they recorded these ERPs while listening to the 2 stories. So this is a schematic of, again, the person lying on their back. The nose is at the top, the ears are on the side. And these circles are the different electrodes that they recorded from. So they recorded from each electrode the pattern of electrical activity in response to the attended story versus the unattended story. And I'm sorry that it's a little hard to see. But what they're showing you is that the attended story is in the dark line and the unattended story is in the dotted line. And this group is all of the average electrodes from the higher maternal education group, and this is all of the averaged electrodes from the lower maternal education group. So I'm going to point out just one electrode to use for an example. So if we look at F3, which is an electrode towards the front of the brain, so underlying prefrontal cortex, and we look at how it looks in the kids whose moms had more education, you see a greater amplitude of response to the attended versus the unattended. The direction doesn't really matter so much, and for conventions that aren't really clear to me, ERP is usually plotted with positive going down. But the point, that doesn't really matter. The point is that you see a greater amplitude of response to the attended, the solid line, versus the unattended, the dotted line. So it's like they have more neural activity to the story they're supposed to pay attention to, okay? Whereas if you look in that same channel in the kids whose moms have less education, you see very little difference across the attended versus the unattended story. And so when we look across all of the electrodes, we see the same pattern. So here we have the attended story on the left and the unattended story on the right. The dark bars represent the moms with more education, or the kids with the moms with more education. The light bars represent the kids with moms with less education. Everyone's generally responding more to the attended story than the unattended story, but this difference between attended versus unattended is much bigger for the kids whose moms have more education compared to the difference for the kids whose moms have less education. So it's almost like they're less able to suppress the distracting information on the neural level. Does that make sense? Okay. Interestingly, the same group a few years later published an intervention study where they recruited a group of kids in Head Start, so that's federally funded preschool for low-income children, and they randomized them to either get Head Start as usual or souped-up preschool, or souped-up Head Start, which was Head Start plus a component that taught children self-regulatory skills, plus that taught their parents different types of parenting skills. So it was, a lot of components, hard to know what the active ingredient was, but basically a Head Start as usual or Head Start with a child and parenting component. And so then they looked before and after the intervention or the control treatment to see what would be the response in these kids' brains to these two different stories. And so the little green filled in part shows you that post-test, so, and PCMCA is their unwieldy name for their intervention. I don't remember what it stands for. But what you see, so basically for Head Start alone, pre-test and post-test, they look very similar, no real difference between the attended and the unattended stories. For the intervention group, pre-test, I think that difference is not significantly different than that. Whereas post-test, you see this significant difference between the attended and the unattended story. So something about teaching kids executive function skills and or teaching parents parenting skills produced better selective attention in these low-income kids. Any questions about that? Okay. Now, executive function and emotion regulation are often considered sort of two sides of the same coin, right? So if we're able to regulate our thoughts and feelings, we're able to somewhat suppress negative emotions, fear, anxiety, anger, right? And our ability to control our emotions is sort of in direct proportion to our ability to self-regulate. And from a neural perspective, emotion regulation is a balance between the amygdala and the prefrontal cortex. So the amygdala is a region we haven't spent too much time talking about today, but it is also very sensitive to stress. And it's a region that detects threat from the environment and activates our physiological stress responses. It has a number of connections with the prefrontal cortex, which is sort of this top-down regulator of the amygdala in adulthood. So as you get more worked up, your amygdala comes online, and then your prefrontal cortex works to kind of calm yourself down. Okay? And we know that increased activity in parts of the prefrontal cortex are associated with diminished amygdala activity in response to negative stimuli, diminished negative affect or negative emotional response, And we know that aberrant prefrontal cortex amygdala regulation is thought to underscore a number of psychiatric disorders, including depression, anxiety, and disorders of impulsivity. So basically, one is sort of your raw emotion, and the other is your ability to keep your emotions in check. Okay. And so one set of researchers, Pil-Young Kim, who's now at the University of Denver, and her mentor, Gary Evans, who's at Cornell, asked whether childhood poverty predicts adult emotion regulation and whether that could be seen on the neural level. So Gary Evans has followed this longitudinal sample of kids who grew up in rural poverty for, I think, a couple decades now. And so at this point, those kids were adults who had grown up and were 24 years old. And so the authors examined the associations between childhood poverty at age 9 and neural circuitry when they grew up to be 24. Specifically neural circuitry during an emotion regulation task that we'll talk about. So they had 49 kids that they had followed on up through adulthood. This was a rural sample in Ithaca, New York. And they had followed them longitudinally every 4 years or so. And they measured chronic stress as a mediator because they reasoned if poverty was associated with these differences in emotion regulation, it would likely be because of these differences in chronic stress that we know have important effects on the amygdala and the prefrontal cortex. So how did they operationalize chronic stress? They called it psychosocial risk. So they measured, sorry about the typo, child and family separation, violence, and family turmoil. They also measured what they called physical risk, so an index of noise, crowding, and housing quality. And they used both maternal and child report, and along the way, some measures of observer report as well. And then finally, at age 24, they measured emotion regulation using what's known as a cognitive reappraisal paradigm. So cognitive reappraisal works like this. You look at a picture like this and you think, ooh, that's a bad picture, right? You have an automatic negative response to that picture. Cognitive reappraisal says, okay, take this negative picture and in your mind, make up a story about why it's not so bad. So, at first, oh, that's so horrible. This toddler is crying. She seems really upset. So if you're making it not so bad, you might say, okay, maybe she just got her shot and she's really upset, but that shot's going to prevent some horrible disease that would have killed her. So it's actually a good story, right? So you could imagine doing that with any number of negative pictures. So what these researchers had their participants do was, in the FMRI scanner, engage in cognitive reappraisal, alternating with just experiencing the natural emotion from looking at a picture. So during the experimental condition, they said, okay, take this bad picture and make it not so bad in your head, decrease the emotional intensity of it. And during the control condition, they said, just experience the pictures in your natural state, let your mind, feel as bad as you want to feel. And so what they asked were, number one, would there be differences or areas of the brain that respond differently during reappraisal versus the control condition? If there are, those might be sort of good candidates for centers of your brain that calm you down, right? And number two, are those differences in those areas related to childhood SES? Any questions about the design? Okay. The answer was yes to both. So they found, number one, that childhood income was related to increased prefrontal and decreased amygdala activation during emotion regulation. So remember, what's happening during emotion regulation? Well, so during an emotional time, your amygdala is ramped up. During emotion regulation, your prefrontal cortex goes up to calm your amygdala down. So what they found was that kids who grew up in higher income homes tended to pull on their prefrontal cortex more during the emotion regulation condition. And in turn, they had less activation in their amygdala. So from a neural perspective, they were better at sort of quieting down their emotions, okay? And that held when controlling for their adult income. So then the next question was, if there were income differences, what might be explaining those? I mean, I told you that they measured different aspects of stress as a potential mediator, right? And indeed, what they found was that this link between higher family income and bringing on the prefrontal cortex when you're trying to regulate your emotions could be explained by how much chronic stress you experienced in childhood. Any questions about that? Okay. All right. So what I want to leave the most time on is how this work could potentially inform interventions. Again, if we have time at the end, I'm happy to go back and talk a little bit about stress in the amygdala or stress in the prefrontal cortex, and also to look at some of the Harvard Center for the Developing Brain videos on resilience, or just answer questions. Okay, so if we're right, and I've sort of tried to paint a picture where we think that childhood experience accounts in large part for these socioeconomic disparities in cognition and achievement, if we're right, then can interventions that target these differences in experience be effective? So that brings us to the sort of fourth part of our linking between SES and the brain, specifically links to achievement and life outcomes. I like this quote by Knudsen: "The most efficient strategy for strengthening the future workforce, both economically and neurobiologically, and improving its quality of life is to invest in the environments of disadvantaged children during their early childhood years." This is work you might recognize that was done right here by Jim Heckman, suggesting that in terms of cost effectiveness, earlier interventions likely have a higher rate of return than later interventions. Not likely, this was an empirical analysis. And so, again, if we're right in the model that we've tried to lay out today, where socioeconomic disparities in experience lead to cascading effects on brain and subsequently cognition, can this work inform interventions? And if so, what is the right level at which to intervene? Okay, so theoretically, you could imagine intervening anywhere along this pathway. And so the question is, which node is most effective? So clearly, we can imagine intervening at the level of cognition itself through school-based interventions. Certainly, that represents the most common form of intervention aimed at addressing socioeconomic disparities in achievement. There are lots of good examples of this. This is work from the Perry Preschool study that many of you are likely familiar with. So this very famous example of extremely high quality preschool provided to low-income children. This was a preschool, entirely staffed by PhD students and their mentors, in which they got really high quality instruction coupled with home visitation. And they've actually followed this sample longitudinally. I think they're now at something like 40 years post preschool. And they've found some pretty remarkable and lasting effects on a number of outcomes. So here in this graph, the kids who were in the control group are shown in red, and the kids in the control group are shown in blue, and the kids who got the intervention are shown in red. So you can see there were reductions in special ed. The number of kids who scored above the 10th percentile is much higher in the kids who got the treatment. The number of kids who are high school graduates is much higher, as is the number of kids who, back when these data were taken, earned $2,000 or more per month, owned their own home, and were never on welfare. So I think in some sense, this is pretty remarkable because it shows the really durable effects of high quality early childhood education. On the other hand, it's certainly, Perry Preschool has been lobbied with a number of criticisms. Anyone, let's say with high school graduation, anything anyone notices about that? So is that actually good? And the difference between the red and the blue is real. What percentage of the kids in red graduated high school? Like 65, right? So that's not actually good. It's better than the counterfactual. There's no question that it made a difference. But it certainly suggests that even like the highest quality preschool that anyone could design is not enough, right? Yeah. Okay. And then, well, we'll talk about some other criticisms lobbied at it as well. Another example of another study that was really thoughtfully done was the Chicago School Readiness Project. So this was a cluster randomized control trial in Head Start settings right here in some of Chicago's poorest neighborhoods. This was led by Sibel Raver and her colleagues, who now she's at NYU. And it was really a comprehensive classroom-based intervention focusing on teachers designed to target emotional and behavioral adjustment. So she essentially trained the teachers, she and her team, trained the teachers in providing support for effectively managing children's dysregulated behavior. So the researchers never provided an intervention directly to the kids. It was all about training teachers in managing problematic behavior in the classroom. And like Perry Preschool, she hasn't followed them quite that long, but she's followed them at least a decade at this point, and probably more. And so what did she find? So as you might expect for a program that was targeting behavioral management and emotion regulation, she did see improvement in kids' executive functioning and their attention and impulsivity. Perhaps even more remarkably, given that there was no emphasis on pre-academic skills at all, was that there were transfers there. So for kids who were randomized to receive the CSRP as compared to Head Start as usual, they actually outperformed their peers on the PPVT, which is a measure of vocabulary, letter naming, which is an early literacy scale, and early math skills. So not only were there effects on early self-regulation, but there was actually transfer to pre-academic skills, even though those weren't targeted, which I think is pretty remarkable. So clearly, school-based interventions can be very promising, but they often suffer from a number of difficulties. So either of these would be considered labor-intensive, right? They certainly tend to be costly if done right. It's hard to imagine, having all preschools staffed by PhDs or their equivalents. And one of the even bigger criticisms is that they often suffer from something known as fade out. So this is this idea that if you measure the results of an intervention immediately after the cessation of the intervention, oftentimes you find large effects. But then if you measure again 6 months or a year or 2 years later, oftentimes those effects fade to nothing. Now, I told you that Perry Preschool, for the most part, has shown somewhat durable effects. They're not as big as they were when they first started, but it still suggested that early preschool leads to some types of long-term effects. The CSRP has had fade out in a number of domains, but not all. Regardless, that's often a difficulty that school-based interventions face. Now, it doesn't suggest that they're not effective, but it does suggest that perhaps they need sort of booster doses of these interventions, which again add to the cost and the labor intensiveness. And then finally, I would say, while I would never, ever deny the critical importance of high-quality early childhood education, if we're waiting until preschool or kindergarten, we're almost certainly waiting too late, right? I've shown you a couple examples now of pretty large disparities before kids even turn 2, right? So if we're waiting until they're 4 or 5 to intervene, we're already well behind the 8 ball. Okay. So what about taking a step back and trying to change children's experience through parenting interventions? I focused on a couple potential experiences that could be targeted today, things like differences in the quality and quantity of language kids hear, things like family stress. And there are many interventions that target those. I'm actually not going to spend too much time talking about those today because in a couple of days, you'll hear from Dina Suskind, whose really wonderful TMW intervention has done quite a bit to encourage families to talk more and more richly with their children. This type of parenting intervention is most commonly home-based, although it doesn't have to be. But particularly early in childhood, it's difficult to get kids within an infrastructure like a school. And again, these can be very effective. But I think as any interventionist would tell you, they also tend to be labor-intensive and costly if done right. And in addition to challenges due to fade out, home-based interventions often suffer from difficulties in terms of uptake and attrition, right? If you're doing an intervention in a school, once the school agrees to let you in, to some extent, you have a captive audience. Now, of course, there could be differences in fidelity. Not everyone's going to do it the same way, but at least you're in. Whereas if you have to go family by family, it's much harder to get people enrolled in your intervention, and it's much harder to keep them there. One notable exception to home-based parenting interventions comes in the form of interventions that take place in the pediatric primary care office, which I think is a really promising future strategy. So something like 90-something% of kids from birth to 3 have a regular source of pediatric care. And so even though they don't go every day, so they wouldn't have the same kind of dose as a school-based intervention, it's at least sort of a home base of sorts where they could frequently obtain intervention services. And again, particularly when you're talking about a home-based intervention, it's pretty challenging to scale up, right? So it's one thing to, do a home visiting program that involves 10 home visits with 100 families. It's quite another to do something like that at the level of the community or beyond. Not to say it can't happen. There are some examples of home-based interventions that really have been done at scale, like nurse-family partnerships, but they're hard and challenging. Okay. And so for the rest of the talk, I'm going to challenge you to thinking about what it would look like to intervene at what I'm calling the most distal level, at the level of SES itself. So you might say, well, you can't randomize people to, or reassign people to different families. That's true. But you can potentially give them cash. So work by my social science colleagues has suggested that relatively small income boosts are correlated with big effects. So for example, correlational work has suggested that a $4,000 increase in annual income between the prenatal year and age 2 is correlated with increased adult earnings when those children grow up, increased time spent in the labor force when those kids grow up, and even some evidence for improved health in adulthood. This is supplemented by data suggesting that increased family income, even in small amounts, is associated with better school achievement in childhood. But those are really all correlational studies. And so we've talked a little bit today about the fact that there's only a limited amount of causal inference that we can make from correlational work. And so we really need to move past correlational work to ask whether income is causing these differences. Put another way, I can show you a ton of evidence to suggest that poverty is associated with detrimental effects on children's development, but there would be plenty of naysayers who would say, well, it's not poverty. It's a mother's ability to pull herself up from her bootstraps. And with correlational work, you really can't say what's causing what. So that really is what spurred the launch of the first clinical trial of poverty reduction in early childhood that my collaborators and I just launched a couple months ago. These pictures remind me to tell you who my collaborators are. So that's Greg Duncan, an economist at UC Irvine. Catherine Magnuson is a social policy expert at the University of Wisconsin-Madison. Lisa Janetian is a behavioral economist at NYU, and Hiro Yoshikawa is a psychologist at NYU. And I feel really privileged to be a neuroscientist working with these 4 social scientists on this study. Although the ambition of the study is pretty large, the premise is actually pretty simple. So we are recruiting 1,000 low-income mothers in the hospital shortly after they give birth. And mothers are randomized to receive either a large monthly cash gift or a nominal monthly cash gift. And they'll be getting that unconditional cash every month for the first 40 months of their children's lives, so just after their children turn 3. It is an unconditional cash transfer, meaning that they can spend it however they please. We're not putting any restrictions on how they spend the money or where they spend the money or when they spend the money. And we're also not encouraging them or teaching them or giving them any financial literacy tools or really in any way trying to nudge their behavior. The treatment group is randomized to receive $333 a month, which comes to $4,000 a year. The control group is randomized to get $20 a month or $240 a year. That difference is an amount that in the correlational literature has been associated with improvements in academic achievement. It's also an amount similar to the earned income tax credit and other social service benefits that are available to disadvantaged families. So it's both an amount that we think is likely to move the needle and also that's policy relevant, right? Even if we could somehow raise the funding to give everyone $50,000, that wouldn't be very policy relevant because it would never be implemented. So upon enrollment in the study, the moms get a prepaid debit card. The card then gets automatically reloaded every month on the anniversary of the child's birthday. And so in this way, we're actually going to be able to move past correlation and assess the causal impact on children's cognitive, emotional, and brain development in the first 3 years of life. Yeah.
Just curious, I know you, especially since you just mentioned things like the earned income tax credit, how is this income going to play into their other benefits that they need to receive?
It's such a great question. So the question you're asking about is something known as clawback. So that's the idea that if I give a mother $333 a month, that may render her ineligible for something like Medicaid or food stamps or WIC or any of another number of other benefits. And obviously that would be terrible, both ethically and then scientifically. It would be a horrible design if we were trying to compare these two groups, but in reality, moms were just losing an equivalent amount of benefits in the one group. So we started planning this study 6 years ago when I was on maternity leave with my oldest, who just graduated kindergarten. And a big part of that was getting this clawback question in order. So we've worked very hard to ensure that moms won't lose other benefits. So we're doing this in 4 metropolitan areas around the country. In 2 of those areas, we've actually gotten legislation passed saying that for these moms, for this study, for this duration of time, this money won't count in determining their benefits. And in the other 2 metropolitan areas, we've actually gone agency by agency to obtain the waivers. So it's not been easy, but we're pretty confident that moms won't be losing benefits as a result. Okay, so what are we actually going to be doing? So at baseline in the hospital, we're administering a survey, and then we're going to be following the families up annually for the next 3 years. At age 1 and 2, we're planning to go into the homes of these families so that we can, in addition to administering a survey, we'll be able to observe and video record parenting and parent-child interaction. We'll be taking some measures of stress physiology from the mom and the baby, doing some basic measures of cognitive development. And then at age 3, we'll be inviting the families into the university lab so that we can do a complete assessment of children's cognitive, emotional, and brain development at age 3. So the study was funded by NIH as well as a consortium of foundations, and we launched this past May. So how do we think that the moms are likely to use this extra cash? We've got 2 main pathways that we're hypothesizing. One we're calling the increased investment pathway. So this is the idea that with more material resources, moms are going to be able to afford more books and toys and trips to the museum, potentially higher quality housing, better childcare, better neighborhoods. And then the other we're calling the reduced stress pathway. So that's the idea that if moms are less worried about making their monthly rent check or keeping the lights on, they have more bandwidth to make decisions about the family, and they're more likely to engage in the kind of warm and nurturing parenting that we know is so critical for children's development. Chances are both of those pathways are operative in different families. And so in a sense, by providing the cash to the families, we're really empowering the families to use the money the way they need best for their own family, as opposed to descending upon them and sort of telling them how to parent or how to use resources. We conducted a pilot study in my lab a few years ago with 30 moms in New York City. And we were happy to find that our procedures seemed quite feasible. So we had over 93% retention over the course of a year. Very few problems with debit card implementation, which was really important for us to measure because for over 1 in 5 of the moms in the pilot study, it was their first time ever using a debit card. So it wasn't at all clear to us that that would unfold seamlessly, but it really did. And I should say that there have been other cash transfer studies that have used other methods of delivering the cash that, for example, might have needed a bank account. So you can imagine the barriers that would potentially be involved systemically with that kind of a setup. But we haven't found that problem with debit card implementation. Okay. Embedded into the pilot study was a qualitative study where we could actually talk to families and try to understand how the money affected their lives. And one theme that we heard over and over was that even in small amounts, the money seemed to make a big difference. One mom told us, believe it or not, even an extra $20 helps. There were times I found myself completely broke. I go and I use it, and that means I can make it for another week. The money from the card really, really helped me out, especially one month that we didn't have the food stamps. We didn't have anything at all. Now, most moms in the pilot study reported using the money for the baby or the other children in the family, but because it's administered by debit card, we, all right, so, okay, so most moms reported spending the mom on the baby or other kids in the family, but because the money was administered by debit card, we don't have to actually take mom's word for it. We can track the transactions. And while I don't expect you to, mentally analyze each of the 1,112 transactions that took place over the course of a year, I do like to point out that there were exactly 3 that took place in a liquor store. Also, other work by social scientists suggests similarly that when low-income individuals have an influx of cash, they're no more likely than high-income individuals to spend on so-called adult vices. Okay. Now, the pilot study wasn't designed to look at the kinds of outcome measures that we're ultimately interested in, so you should certainly take this with a grain of salt. Nonetheless, of course, we took a little peek at our 1-year outcomes, which was the duration of the pilot. And despite the fact that it was a very small sample, just 15 in each group, patterns on the order of half a standard deviation favored the treatment group in terms of higher reports of center-based childcare expenditures, more frequent mother-child activities, less household chaos, and less parenting stress. Now, again, we didn't measure cognitive development in the pilot. It was really mostly a pilot to test feasibility. So the real study is where we're going to be able to look at those types of outcomes, but at least suggest some promise. Yeah.
I talked a lot about moms. I'm just curious, are you also looking at, are these single-parent households, or dual-parent, and have you also looked at like the dad's implications or these?
Yeah. It's a great question. So we are taking all comers in terms of single parents or any other family structure. We are recruiting the moms in the postpartum ward. So the moms are sort of the point of entry, and they are the one given the cash, but of course they can share it with whomever they would like. We are asking, so at the subsequent rounds, we're asking about father involvement, father figure involvement, and the involvement of other family members. We do not currently have plans to directly assess the father just because of limitation in time and resources. But you could imagine that kind of supplemental study specifically measuring the fathers. But right now, any information about fathers is obtained through the mother.
So of the 15, did some of them have a father figure in their life? Were some of them married? Were most of them not married? They were moms?
I don't remember of the 15. Certainly were, I mean, yes, there were some of both, but I don't remember the numbers. And certainly in the larger study, we will see, all types. So we're not constraining by family structure. We do have a plan. So about 40% of births nationally are first births. And so you could imagine, a lot of times we get asked whether we're limiting it to first births or not. So you could imagine that the money would go farther in a first birth. On the other hand, first births often have younger moms. They might be less, used to budgeting their finances. So you could kind of imagine it going either way. We've decided to constrain ourselves to no more than 50% first births and no more than 10% of parity of 4 or more. But we'll sort of take all comers until it turns out that we have to differentially sample. It's sort of purposefully left, oh yeah, go ahead.
I'm sure you get this all the time, but are you tracking labor force participation in hours?
Yeah, it's such an important question. So yes, so are we tracking labor force participation, right? I can tell it's a group of economists. So a lot of people would say if you give cash, that's going to offset work, right? And so is that something that we want to do? I guess there's the sort of question on the larger level of whether offsetting work is a good or a bad thing. Certainly you could imagine that the net income gain is 0 if a mom chooses to offset work. I think one important thing to keep in mind here is that we're talking about the effect on children in early childhood. And so in some sense, it's going to be very applicable to paid family leave models. And so from a developmental perspective, I think offsetting work and keeping income the same is likely a very good effect for children at this age. I mean, it's an empirical question, and we'll be able to find out the answer. Other questions? Okay. So if our hypotheses are right, and boosting family income can change children's trajectories for the better, it could have some real policy implications. So currently, the United States pales in comparison to our developed country peers in terms of the amount of public spending on children and families. But it doesn't really have to be this way. So earlier I showed you some data looking at rates of poverty over time. So here I'm showing you the data again, presented slightly differently. In blue, you see senior citizens, and in red, you see children. And so what you see is that back in the late 1950s, the rates of poverty for senior citizens were actually quite high, over 1 in 3. But with the advent of things like Medicare and Social Security, those numbers have steadily dropped to where they are today, affecting fewer than 10% of senior citizens. In contrast, with children, back in the late 50s, we were at 27% of childhood poverty. Now that number decreased a little bit with the advent of Medicaid, but because spending on children in poverty has not traditionally been a priority of the government, the number has steadily risen to the 1 in 5 where we are today, and really where we've been somewhat steadily for quite some time. So again, this is really just an illustration of the fact that it doesn't have to be that way, that governmental priorities can shift and can have very real ramifications for rates of poverty in different demographics. So we think that this study has the potential to provide direct evidence of the effects of poverty reduction on the developing brain and mind. We're hoping that it will also inform debate on the generosity or cuts both to existing social service programs as well as potential new service programs that will affect the lives of millions of disadvantaged families with young kids. So things like SNAP or food stamps, WIC or the Women, Infant, and Children program, TANF, which was formerly known as welfare, housing vouchers, paid family leave, and the minimum wage. And so while I would never suggest that income is the only or even the most important factor in children's brain development, it may be the most manipulable from a policy perspective. So that represents the end of my formal slides. Go ahead and follow me on Twitter if you'd like. I'm happy to take questions. If there are no questions, we can go back and take a look at some of those videos I mentioned, or talk a little bit more in depth about the neuroscience of stress in other brain circuitry, but I thought there might be some questions either on this study or broadly what I covered. Yeah.
So I know you said in this pilot study that you all did, there were no instructions on how to spend the money, there were no additional support.
And there won't be on the larger study. Yeah.
Yeah. Do you think there could be potentially, higher benefits if there are additional support?
If we did,
provided?
yeah. So potentially, right? So the study as it is now is really designed to be a pure test of income. Does income in and of itself lead to differences in child development? Which is, I think, is a sort of reasonable starting point. You could certainly imagine it paired with any of a number of other interventions, parenting interventions to improve language exposure, to reduce stress, to, reduce violence, to change lead levels, to increase financial literacy, right? And so at some level, it's a question of which intervention would you pair with it. If we're right and we do find differences, then you could certainly imagine subsequent studies that, pair cash with other things that cost more or less, right? Because one, I think, key question, if we do find differences, one key question might be about cost and benefit, right? And how much does $4,000 a year of pure cash get you as opposed to $4,000 a year spent on a program?
I'm curious if you can talk more about, so I do child welfare work, and I'm thinking a lot about adverse child's experiences and research around that. And the prevention stuff with SES is really, really cool, but I'm curious if you know anything more about interventions for once kids have experienced adverse experiences, or child welfare involvement and things like that. What do we do to help them when they're in these positions where their brain development is reduced and they're older? Is there any hope for those groups?
Yeah, there are. So I'm thinking of work by people like Mary Dozier, who've done a lot of work, and Phil Fisher, who've done a lot of work with foster care interventions. So teaching foster parents about different ways to parent. They're labor-intensive interventions, but very beneficial and I think very important for those kinds of targeted groups that really need substantial extra services. So, for example, foster parenting is very challenging, as you might imagine, for lots of, as you know, for lots of reasons, right? So, for one thing, kids in the foster care system often had antecedent trauma or other issues leading to behavioral difficulties. So these foster parents are getting kids who are already primed to have difficulties with behavior regulation. Certainly not always, but often foster parents themselves had issues becoming a biological parent, and so they're often going into that situation with their own baggage. And then coupled with those two sort of separate child and foster parent factors is the fact that often, despite a foster parent's effort, they're sort of met with these difficulties of the child. And so they're really not getting the kind of positive feedback you'd want them to get, and they often internalize it or shut down. And so it can often be a pretty vicious cycle where the kids in foster children, despite the good intentions of the foster parent, aren't getting the kind of extra warmth and nurturing you'd want them to get. And so Mary Dozier's intervention, for example, is a home visiting program that I think visits families 10 times, and she does this videotaping. She does do videotaping, but they also in the moment kind of coach them. They're supposed to give them, I forget how many little pieces of feedback every minute, where little praise for good things that the foster parent does in the course of interacting with the kid. Then they watch the videos afterwards and kind of go over it. And they see real change in parenting and subsequent child development, and actually child stress physiology following the intervention. Now, in some ways, her intervention has been scaled. I mean, it's been picked up by a number of foster agency systems and other communities around the country. But she's maintained very tight control over it, understandably, because she's worked very hard to develop this wonderful tool. But for a program to adopt her intervention, they need quite a bit of training, and they need, over the first year of implementation, they need to send videos back to her lab to be coded and sort of make sure that there's no drift. So they keep tight control over it, which is awesome. But on the other hand, that's only scalable to a point, right? And not every foster intervention can run itself through Mary Dozier's lab. And so I think that is the inherent challenge between high quality interventions and scalable interventions. Yeah.
I love learning about concepts that you're like, oh, that makes sense because of this. And we just had a student change from an ODD, oppositional defiance disorder, to a DMDD, and we were just really disturbed just because his diagnoses keep on changing, which affects med, which affects lots of things. And so the cognitive reappraisal you were talking about is so helpful. I just feel very grateful that what that would be for him would be that we can reframe things for him, saying that they're really working hard, they're really going to tie in, but also, I guess your example not only acknowledged kind of the dysfunction, but then kind of reframed it, as opposed to, because if both aren't present, then it's not going to work, right? The acknowledgement has to happen and the reframing, and not just the Pollyanna reframing, and not just the acknowledgement and the negative Nelly together thing. And so is cognitive reappraisal being used for even for example for the anti-suspension measures in K-12, like the SB 100 bill with negative tracking between African American boys and how many suspensions there are? Are they using kind of some of this cognitive neuroscience for the educational policy stuff, or is there still too much of a disconnect between the two worlds?
It's a great question. So I don't know specifically for that example, but I do know that it is used as a technique in cognitive behavioral therapy, right? So as a therapeutic technique for sort of reframing your world. So it definitely does have therapeutic approaches. You can look up the work of Kevin Oaksner at Columbia, who's done a lot of the neuroscience of cognitive his last name is spelled, I think, Ochsner. Anyway, he's in psychology at Columbia, and he's done a lot of the neuroscience work of cognitive reappraisal and would likely reference the clinical work in some of his work as well. Of course. Yeah.
Quick question. I think it relates to the question prior, just in terms of, is it too late? Do you have any thoughts around sort of the critical periods and the malleability of that, and the research being done at Harvard?
Yeah. So thoughts on the critical periods and malleability of the developing brain. So I can say, I don't think there's a hard cutoff. We all know that we continue to learn as adults, right? And even things like a second language are learnable in adulthood. That said, the general consensus does seem to be that we are more plastic. We are better at learning early in childhood. One classic example comes from the literature looking at second language acquisition, and it's much easier to learn a second language early in childhood than it is later in childhood or in adulthood. It's not to say it's impossible later, but almost always will be accompanied by an accent, and even later than that, almost always accompanied by grammatical errors. whereas that's virtually eliminated if you teach the second language earlier. And that seems to apply to the early adversity literature as well. I don't think there's like a hard cutoff.
Takao Hench's stuff, his research.
Yeah.
You know about where
Yeah,
sort of
okay.
molecularly changes the critical period?
Yeah, no, that's a great question. So let me do BEIP first, and then I'll talk about Takao Hench as like a positive. So the Bucharest Early Intervention Project you may have heard about, that's a study that looked at kids reared in Romanian orphanages. And and so this is sort of the extreme of deprivation. And the researchers randomized kids to either receive care in foster care in Romania or care as usual, so to continue to be raised in the orphanage, in the institution. And it looked like there was a timing effect. It continues to. They continue to follow these kids up to this day. And they're now in their late teens. So it looks like there's a timing effect whereby the kids who were adopted into foster homes before the age of 2, not always, but often look normal, and at a minimum, they always look better than the kids who were adopted after the age of 2. So even for kids who were adopted out, if it happened a little bit later, it seems like there was some kind of critical period, or at least sensitive period there, where the window is before age 2 to sort of buffer against the effects of early adversity. Takao Hench has done some really interesting molecular work looking at re-inducing plasticity pharmacologically in adulthood. So it's certainly still experimental, but there seems to be some indication that perhaps with pharmacology, with drugs, we could re-induce plasticity even when it looks like that critical period has closed. Now, we are very far from answering the kinds of questions you might be thinking about, can we reverse the adversity of orphanage rearing? I mean, we just don't know. Certainly no one's tried that kind of work in humans, but that's where sort of the frontier of the field is in terms of thinking about plasticity. Does that answer your question? Yeah.
I'm really curious, I know we've gone back and forth on education of the mother versus income. I'm still just a little unclear on, or if you have opinions on why sometimes one matters and why sometimes the other one matters.
Yeah, it's very unclear. So, and I think your non-clarity on it is reflective of the field at large. So people measure all different kinds of things. Sometimes you see maternal education, sometimes you see paternal, sometimes you see a composite of education and income, sometimes you see them separately. So following my reading of and collaboration with social scientists, I've taken the tack of measuring everything and considering them separately, because even though education and income are often correlated, they seem to reflect different underlying processes in the child's life. At least that's my take on the social science literature. And so I don't have the greatest answer for you. I mean, I wish I could tell you cleanly, education seems to be more related to parenting and income seems to be more related to material resources and financial stress. That's like broadly true, but there are plenty of examples where that doesn't always hold. So I don't think that there's a clear line, and it's probably because those are both distal measures of family processes, right? And so, is generally having a higher education associated with talking more richly with your kids? Yes, but it's not a one-to-one correlation.
And have there been any others that have been added or looked at? So perhaps like two-parent households or grandparents are involved. I don't know. Is there anything else that?
Yeah, so as you would imagine, single parenthood tends to provide risks for negative outcomes, and involvement of other family members tends to buffer against those risks. But again, we're talking about sort of population level risks, and it's very hard to generalize to an individual. Sure. Other questions? One thing about thinking about the question about scalability, so when we think about the cash transfer study, it's sort of an interesting question about what constitutes an efficacious intervention, right? So an efficacious intervention on the one hand is one that moves the needle a whole lot, right? Like makes a big difference in outcomes. On the other hand, from a public health perspective, it also needs to be an intervention that has high uptake. So, if you think about something like preschool, made a big difference, but it's really hard to implement. And so you're not going to see a whole lot of preschools at scale that are that sort of high quality preschool, right? It's just not, there's no funding to make it be so. Giving people cash is something that, so I say it sort of facetiously, it's really easy to do because it's actually been a very long time in coming, but when we're actually at the point of offering it to people, people tend to say yes, right? They take the cash and they use it, they spend it quickly. So in terms of uptake, it's nearly universal. Now, the jury is still out, right? It's an empirical question as to how much will move the needle. And you know, is it likely to have the same effect, in my opinion, is it likely to have the same effect as, you know, the highest quality preschool with all the bells and whistles? Probably not. But if it's nearly universal uptake, then even a relatively modest difference would still be a very large public health impact. And I think that's sort of an important thing to consider when you're thinking about the public health impact of interventions and the reach that they're likely to have. Other questions? All right, why don't we watch one of those videos, because I think that they're a nice resource, and they sort of do a very good job explaining the role of toxic stress.
Learning to deal with stress is an important part of healthy development. When experiencing stress, the stress response system is activated. The body and brain go on alert. There's an adrenaline rush, increased heart rate, and an increase in stress hormone levels. When the stress is relieved after a short time, or a young child receives support from caring adults, the stress response winds down and the body quickly returns to normal. In severe situations, such as ongoing abuse and neglect, where there is no caring adult to act as a buffer against the stress response stays activated. Even when there is no apparent physical harm, the extended absence of response from adults can activate the stress response system. Constant activation of the stress response overloads developing systems, with serious lifelong consequences for the child. This is known as toxic stress. Over time, this results in a stress response system set permanently on high alert. In the areas of the brain dedicated to learning and reasoning, the neural connections that comprise brain architecture are weaker and fewer in number. Science shows that the prolonged activation of stress hormones in early childhood can actually reduce neural connections in these important areas of the brain at just the time when they should be growing new ones. Toxic stress can be avoided if we ensure that the environments in which children grow and develop are nurturing, stable, and engaging.
Why do some people do well despite serious hardships? The science of resilience can help us find answers.
We define resilience as a good outcome in the face of adversity.
The extent to which we can build capacities in all children early in their lives to be able to deal with whatever bumps in the road or major obstacles may be coming down the track. That's an investment in building strong human capital and healthy, productive adults.
Some children face more obstacles and adversity than others.
Like having a parent with a major mental illness, growing up in a very socioeconomically disadvantaged community, going to schools that are not good, being exposed to violence in the neighborhood or in the home.
But some children do well despite these significant obstacles.
Resilience is that ability or set of capacities for positive adaptation, allowing you to keep in balance.
We're talking about the kinds of capacities and skills and the abilities that give people a sense of mastery and management of difficulty.
It's tempting to think about children as either having this resilient quality or not. But resilience is built over time, just as and in parallel with how the architecture of the brain is built over time.
It's not just in the person. It's in the interaction between the person and the environment.
So we care about resilience for the same reason that we care about promoting healthy development, because in many respects it is the same. We are interested in promoting resilience in children so that, despite the odds, more and more children can grow up to be productive citizens.
So that was good, but that actually wasn't what I was looking for. I mean, they have a whole video about it, but I could just sort of explain it. So at the Center for the Developing Child, they conceptualize resilience as sort of being this scale with a fulcrum, right? And so you've got your positive and negative experiences weighing you on one side, but your fulcrum is sort of like your genetic predisposition, right? So different individuals are genetically predisposed to weight more or weight less their negative experiences. So in some cases, a little bit of negative experience can lead to a bad outcome. In other cases, it takes a whole lot of negative experience to lead to their whole outcome. So it sort of depends where that fulcrum is, and that's this conceptualization is based on genetic predispositions moving the fulcrum. But even if the fulcrum is in a very disadvantageous position, it's still the case that with enough positive experiences, you can sort of overcome where that is, the fulcrum is placed. So which I think is a great metaphor for kind of how both genes and experience are important and how genetic differences may predispose one to differences, but experiences can sort of modulate or change the effect of those genes. In some ways, that's actually the basis for epigenetics. I don't know if you guys are familiar with that term. So the idea is that, we're all born with our DNA. That part doesn't change. But what does change is the extent to which our DNA is turned on or off. Okay, so that's where epigenetics comes in. And so our experiences play a role in determining which genes are turned on or off and how frequently. And there's quite a bit of work looking at early adversity and epigenetic modification, and even cellular aging, right? So there's some, I told you earlier that there's some evidence that brain development seems to be accelerated in situations of early adversity. Maybe that's adaptive, right? Maybe you want to be more adult-like if you're in an adverse situation so that you can become more independent more quickly. There's some suggestion of early bodily aging as well. So, that early adversity actually speeds cellular aging, and so some question as to whether interventions could potentially reverse that and actually slow biological aging. It's something we're hoping to look at in our RCT. Did you have a question?